Okay, so I'm going to go through the questions that people put on the discussion board on Blackboard. So thank you for doing that. And I'm going to go in the order that they were posted. So our first question is, can you explain how Page and SDS Page are different from each other? So let's go to that. And I'm gonna pop up a whiteboard so I can write while I do that. So we're looking at Page versus SDS Page. And the difference, which is a big difference, is SDS, which stands for sodium dodecyl. That's not how you spell it, but it's close enough. Dodecyl sulfate, which is a detergent, and it's negatively charged. So what SDS Page, or what SDS does when it's exposed to a protein, it does two things. Unfolds the protein and gives it uniform negative charge. So Page stands for polyacrylamide gel, electrophoresis. So in both of these techniques, you have a gel that's made out of acrylamide, and you use electricity, where you usually have the positive end down here and the negative end here. So in SDS Page, since everything's unfolded and everything has a negative charge, the proteins only separate by size. However, if you're just doing native Page, there's a couple things you have to, well, there's more variables, because the protein is folded in native Page, and it doesn't necessarily have a net negative charge, because the charge of the protein is based on whatever amino acids it has. So in Page, you separate by size, you separate by shape, and you separate by charge. So you might ask yourself, why would I do Page versus SDS Page if I have so many variables? Well, with Page, what you can do is that you can actually run a sample through Page, then extract, like if you get a band in Page, you can take a knife, extract that, remove the acrylamide, and you still have folded usable proteins. So it's a way to separate proteins while still having them usable and intact. And also, if you're doing a test with antibodies, and the antibody only works against the folded protein, you would have to do Page to detect your protein. Otherwise, SDS Page is better because you're only separating things by size. And the other part of that question is, could you go over the question from the 621 lecture? And that question just says, you're giving 4 proteins, rank them from how far they will travel down the gel. So the 4 proteins are 32 kilodaltons. So protein 1, Protein 2, 135 kilodaltons. Protein 3, 85 kilodaltons. Protein 4, 55 kilodaltons. And in SDS page, you're separating by size. Small things are able to go through the gel faster. So it'd basically be protein 1 would be down here, protein 4 would be down here, protein 3, and then protein 2. So small things travel through the gel faster than big things. So that is SDS page versus page. Let me go to our next question. And our next question is cation exchange. Can I go over the question on the exam with cation exchange? So let me clear this and rewrite the question here. So we have cation exchange, which means we bind cations. We're exchanging those. That's what we bind. And we have 4 proteins, A, B, C, and D. pKa of 2.4, molecular weight of 24. These are, all the weights are in kilodaltons. B, pKa of 9.5, weight of 96. C, pKa 12.5, weight of 33. D, pKa 10.6, weight of 17. Now, the weights in this problem aren't used. They're just there to throw you off and make sure that you actually know what cation exchange is. So they're completely meaningless in this problem. All we care about are pKa's. And for proteins, the way it goes is that low pKa's means you have a lot of negative amino acids. High pKa's means you have a lot of positive amino acids. And we already talked about this, but the idea is if you remember like your pKa's like D and E, you're looking at like a pKa of like 3.5 and 4.25, so low numbers. And remember, when the pKa is equal to your pH, that's when you become neutralized. You're 50% charged, 50% neutral. So if a protein has a very low pKa, and for proteins, the pKa is where you're neutral. So for example, if protein A is only neutral charge at a pH of 2.4, that's what this means, then it must have a lot of negative amino acids, because it needs all the protons in solution to protonate those negative amino acids and become a neutral molecule. So in cation exchange, then, we are binding cations. So the first thing to come out of this column would be protein A. That's the first to elute, because it's all negative, right? Very negatively charged. So it's not going to bind to the column that strongly. Then it will be protein B, D, and C. And the reason for that order is that protein C has the highest pKa. So it's the most positively charged protein. Since it has more positive charges, it's going to bind to that cation exchange column stronger, and therefore be harder to come off the column. So just keep these couple of things in mind, right? Low pKa, if you're a protein, means you have negative amino acids. High pKa means you have a lot of positive amino acids. And just remember cation, anion exchange. Cations bind cations, anions bind anions, right? And cation are positive, anions are negative. So if you have that information, I think you can do the ordering quite easily after that. And if you have questions during this, feel free to just chime in the chat box. Otherwise, I'm going to be going to our next question on the message board. And that question says, how will the final exam be structured? Will it be 25% over new material and 7% over the old material? I would say it's closer to 10 to 15% new material and 85% old material, because it's going to be, because the new material isn't a quarter of the class, right? The new material that we haven't been tested on on our last test, that will be all new for the final exam. That's 10 to 15% of the class. So that's going to be 10 to 15% of the final exam. It's roughly going to be representative based on how long we spend on different topics, to, you know, as a rough estimate. And another question, will we be tested over sections 12.3 and 12.4 or just up to section 12.2? Just up to 12.2. 12.3, 12.4 was the stuff for today. But since I decided to make this review instead of going over that, I'm not going to hold you accountable to that on the final exam. So just up to what we covered on Wednesday is going to be what your final's over. All right, our next question here is inactivation ball. What causes the inactivation ball to swing in and physically obstruct the pore? Slide 7 from 726. So let me get that open so we can take a look at that. And this goes with the potassium channel. Oh, let me just share that PowerPoint. 726. All right, so if you remember let me get my pen back up. There we go. So the potassium pore has these positive amino acids on helix 4. Sodium will flow into the cell, which will make these positive charge move away from the inside, opening the pore. And the way that this pore closes is that on the end terminus, there's called an inactivation ball that swings right into the pore and closes it. And the reason why they interact is because this inactivation ball is hydrophobic amino acids. And at the very opening of the pore, you also have hydrophobic amino acids. So when this pore is open, these hydrophobic amino acids are exposed to the cytoplasm. And you have this very flexible end terminus that's near the pore that also has hydrophobic amino acids. So it's the hydrophobic effect. Water will push these hydrophobic amino acids to interact with the pore's hydrophobic amino acids, and this physically seals the pore, so potassium can no longer escape out of the cell. So yeah, that's how the inactivation ball works. It's all hydrophobic interactions. Let's see what else we have here. Next question is about hyaluronic acid. And can I explain why how hyaluronic acid is a heteropolysaccharide? Let me get that slide up. So here is hyaluronate, which will make hyaluronic acid. And let's break down that word, heteropolysaccharide. So hetero means different. Poly means many. Saccharide means sugar. So heteropolysaccharide means many sugars that are different. So hyaluronate is made out of 2 sugars, glycouronate and N-acetyl D-glucosamine. That's where the hetero part comes from, because they are different. And this compound will just be these 2 disaccharides connected, just repeated over and over and over again. So you'd have like 2, 2, 2, 2, 2, all connected together. So that's where the poly comes from, because there's many of these disaccharides linked together. And saccharide, because it's a sugar. These are sugars. So all of these compounds I'm actually showing on this slide are heteropolysaccharides. They are each made out of different monomer units, and they repeat over and over and over again. Our next question is about antigens. Could you talk more on the carboxy terminal of an antigen? The antigen question on test 3 confused me. So I believe you mean antibody, because let's do some terminology here as I open up the test. And go to antigen. All right, let me clear this and share the test. All right. So first, I just want to start with some terminology. And whoa, did not want to draw that like that. All right. So this is an antibody. And whatever an antibody binds is an antigen. All right. So an antigen is the thing an antibody binds, and it can be literally anything. So when we're talking about antigen binding sites, we're talking about antibodies. Like what is, yeah, they're just saying where on an antibody will you bind an antigen? So we need to look at which statement's false. And how best to do this. So what I'm going to do is I'm going to open up the antigen slide and then read off the questions and tell you why they are true or why they are false. So again, I'm going to switch screens on you so we can look at the antibody here. And let me get the question back up. All right, so our first statement, an antigen binding site on an IgG is just a antibody. What you're looking there, that Y shape, is an IgG, immunoglobulin G. You have a bunch of different types of antibodies, immunoglobulins in your body, and they're just lettered based on different classes. So we're just looking at IgG. Right. So an antigen binding site on an IgG is formed by the amino terminal 110 amino acids of the light chain, and the amino terminal, 110 amino acids of the heavy chain. If we look at the antigen binding sites right here and right here, and this is the N-terminus of the antibody of the heavy and light chains. So you have two different chains, light and heavy, right? The C-terminus of the heavy chain is right here. The C-term of the light chain is right here. And these are the N-termini. And both the N-termini of the heavy and the light chain make up where you bind the antigen. So that's a true statement. So that statement's true. Our next statement, an antigen binding site on an IgG is formed by the variable region of the light chain and the variable region of a heavy chain. That is true. The parts in red here, let me erase my circles. These are the variable regions. That means that the amino acids there are different in every single antibody, because you need to be able to bind any possible antigen you come across inside the body. So these are variable, they change amino acid composition, and that is where you bind antigen, so that's true. The antigen site is composed of two Ig folds. That is true. What an Ig fold is, it's a type of structure, and it's actually on this slide. And let me erase that. So the immunoglobulin fold is this beta sandwich, and it's called a beta sandwich because you have two beta sheets that are on top of each other, like two buns on a sandwich. And this is, these are the parts that are in the antibody. So you have one in the hypervariable region and also constant region. So what you're looking at here is, let me go back to this slide. So we're zoomed in like on the light chain when we're looking at the IgG folds. So keep that in mind. That's what we're looking at when we go to this picture. Or this is C-terminus and N-terminus. So yes, it is made out of IgG folds where we bind. So that is a true statement. And the next statement is antigen binding specificity is determined by the sequences of the hypervariable sequence in both the light chain and heavy chain. That's true. And it kind of goes back to our previous statement, but both the heavy chain and light chain, that's where you get VH and VL from. Light, heavy, variable, variable. This is where you're binding the antigen. So yes, it's both the heavy and light chain that make up the binding site, and they're both variable. Then our last statement is the false statement. Antigen binding specificity is determined exclusively by the sequences in the carboxy terminal, 110 amino acids in light chain and heavy chain. So that's false. The carboxy ends of the light and heavy chains, the C termini, they are all constant. Every single antibody has the same sequence in the constant region. So these do not bind antigens. They don't change. It's the N termini. So that was the false statement there. So let's go to our next question. And it's about active transport in the sodium-potassium pump. And it's what drives the E2 potassium complex back into the cell? Is it due to the hydrolysis of a phosphate that results in a conformational change to E1? So just a little clarification there, and I can stop sharing the antibody and I can share that question. as I'll be talking about it. All right, so just some clarification. The enzyme itself isn't moving outside and inside of the cell, right? Even though that the image on the PowerPoint looks like it is physically moving, the enzyme is in the membrane, the cell membrane. So the enzyme just stays in the membrane. So what is driving the potassium? Yeah, that's the energy of ATP. When that ATP is being hydrolyzed, it changes conformation. And let me just go, let me see if I can't pull up that slide. So I don't think I have that up right now. Yeah, okay, there it is. So let me share that. Stop sharing this and share this. So the question is, why is that potassium being pumped into the cell? And it is, so if we go back a couple steps, right? In the E2 form, you have phosphate bound, and that's a stable conformation. And then potassiums will bind to that. And once that phosphate is removed, this becomes unstable. That's what this this star kind of means. That star is saying this is a high energy form. So it does not like to stay in the E2 form without a phosphate. That the protein is high energy, unstable. And so the lower energy conformation is E1 with ATP. So ATP can bind to this E2 and turn it into E1. And when it does that, potassiums will flow out. And it doesn't happen at the same exact time, but it kind of does. What I mean by that is that if we were to look at the mechanism, we would probably see, ATP bind. We would probably then see a conformational change. And then we would see the potassiums dissociate from the transporter. But that happens so fast that it looks instantaneous to us. But yeah, that's what's going on. You remove the phosphate, this enzyme becomes in a high energy state. Molecules don't like to be a high energy, so it's going to go to an E1 state that's lower energy, and ATP will bind to the E1 state, and that's causing that energy going from E2 to E1 is what is the conformational change that is on that potassium to leave. All right, next question here. Can I provide a quick definition of secondary active transport? Sure. Let me get the whiteboard back out so I can draw this and clear that. So we talked about secondary active transport when it came to the sodium glucose system, the SYMPORT, where we had glucose come in and we had sodium come in. So the movement of glucose into the cell is unfavorable, energetically speaking, but the movement of sodium is favorable. So secondary active transport means we're using active transport, but not ATP. Here we're using the electricity gradient of sodium, right? Sodium is moving into the cell. The cell is negatively charged inside. I believe it's I don't know the concentrations of sodium inside and outside the cell off the top of my head, but I know this is a moving sodium from outside the cell inside the cell is an overall favorable process. And so we're using that energy to move glucose, which is an unfavorable process. So that's a quick definition of secondary active transport. It's active transport, but you don't use ATP as your energy. You use an electrochemical gradient. So sodium going into the cell is a favorable process. So we're using that energy to move glucose. And if you remember, the whole process is now glucose can go into the bloodstream. So this is in your small intestines where this is happening. Glucose can go in, but for sodium to leave, and this is actually a sodium potassium pump. Here we're using ATP because they pump sodium outside the cell. If it's favorable going in, it must be unfavorable going out. So here we are using ATP in a second process. But secondary active transport itself is just using an electrochemical gradient. And we do actually use ATP in a separate but related process here. So that's secondary active transport. Right, our next question is relating pH and PI or pKa of proteins. And they posted a question on the exam. So let me rewrite that question. So the question is, which of these peptides is positive at pH 7? I have LKG, REM, ATN, QVS, PIL. All right, so this one, positive at pH 7, there's actually no calculations you have to do. This was a straight, do you know your amino acids, right? So at a pH of 7, your negatively charged amino acids are D and E. Your positively charged amino acids are R, K, and usually for these calculations, we say H is positively charged. Hopefully, now at the end of the semester, you've learned that, there's an asterisk to that where it's really, 60% of the time it'll be charged and 40% it won't be. So it's really a toss-up. But for these calculations at a pH of 7, we usually say, yeah, just treat H as positive. It doesn't matter for this question though, because there is no histidines. All right, so let's look at this. So leucine, pH 7, neutral, because it's not on our list. Lysine, positive. Glycine, neutral. Okay, this has a positive 1 charge. Done. That's positive at a pH of 7. We can go through the other ones though. REM, R is positive, E is negative. M, not one of these 5, so it's neutral. So this has a charge of 0. A, neutral. T, neutral. N, neutral, neutral. Q, neutral. B, neutral. S, neutral, neutral. P, neutral. I, neutral. L, neutral, neutral. So that's that question, but I'm going to expand this a little more because I do want you to know the charges of peptides at different pHs. So let's look at this. We're going to look at LKG. So remember, every peptide has an N-terminus and C-terminus. The pKa of the N-terminus is roughly 9. The pKa of the C-terminus is roughly 2. And K is roughly 10.5. L doesn't have a pKa. G doesn't have a pKa. So if you don't have a pKa, you're going to be neutral. So the type of question I like to ask for this is, what is the charge of this peptide at pH of 1, pH of 7, and pH of 14? So we just did 7. So let's do that. So the N-terminus at a pH of 7 is neutral. Lysine, and let me make some lines here. Treat this like a table. And then the N-terminus like that. All right, so the N-terminus is positively charged. Leucine, neutral. Lysine, positively charged. Glycine, neutral. Cysteine, C-terminus, negatively charged. Overall charge, positive, positive, negative. So +1, +2, -1. So overall +1. I'm just adding up the charges there. All right, pH of 1. Basically, at a pH of 1, anything that's negative becomes neutral. And the reason why this is that if we look at the C term, that has a pKa of 2. At a pH of 7, you are in this structure, right? COO, O-. So you're negatively charged. At a pH of 1, you're now below the pKa. You're at a pH lower than the pKa, which means you are now going to be protonated, right? When you go below your, when the pH is below the pKa, you become protonated. So this becomes OH. And so at a pH of 1, the C-terminal is neutral. Glycine doesn't have a pKa, so it doesn't change. Leucine doesn't have a pKa, so it doesn't change. So let's look at lysine now for an example. So lysine has a pKa of 10.5. And the very end of the lysine is an NH3, which is positively charged. And so at a pH of 7, we're already below the pKa of 10.5. So we're already protonated at a pH of 7. So if we go to a pH of 1, right? Oops, NH3. We're still below 10.5. So nothing changes. Like you don't lose another, or you don't gain another proton. If you're still below the pKa, nothing in your structure changes. So lysine still remains positive. Same thing for the N-terminus. So overall, at a pH of 1, the charge of this peptide is +2. Now let's go to a pH of 14. So lysine, no pKa, doesn't change. Glycine, no pKa, doesn't change. The C-terminus. So like we were saying, the C-terminus is negative charge at a pH of 7. So going to a pH of 14, we're still above the pKa of 2, so it doesn't change, right? It's still negatively charged. However, lysine at a pH of 14, we are now above the pKa. When the pH is above the pKa, you lose a proton. You become deprotonated. So this NH3 group becomes an NH2. So lysine is neutral. The N-terminus, likewise, is neutral. So overall, at a pH of 14, this peptide has a charge of -1. So some things to keep in mind. If the pH is above the pKa, you lose a proton. If the pH is below the pKa, you gain a proton. Amino acids are either +1, neutral, or -1. You can't have an amino acids that's +2 or -2. That's a common mistake I see with this, where people try to say, well, okay, for example, for lysine, at a pH of 1, a lot of people want to say lysine is +2 because you're below the pKa, so you must gain a proton. But lysine was already positively charged at a pH of 7, so it's not going to be +2. You would have to go to a pH of like -10, -30 to make lysine +2. You just have to go to ridiculous pHs. So keep that in mind when it comes to determining the charge of peptides. The relationship between pH and pKa, whether you're going to gain a proton or lose a proton, and know your pKa's of your amino acids makes this problem a lot simpler. So that was that excuse me. That was that problem. Let's go to what else is on the message board. All right, so this question is about transition state binding. Since enzymes want to bind the transition state of a substrate, why are they okay with the transition state continuing to products? And what is the entropy penalty of the enzyme keeping substrates in the transition state? All right. So for this, I'm going to draw our reaction diagram. So energy on the Y, reaction progress on the X. So substrate, product, non-catalyzed, top line, bottom line is catalyzed reaction. And this bump is the transition state. So the first question is, enzymes really want to bind the transition state. They are made to do that. So why don't they just stay in the transition state? And that's all due to energy, right? Even though they want to bind the transition state, it's still the most high energy compound. It's still the compound in this whole reaction that is the most unstable. So naturally, molecules want to go to low energy states. They want to be the lowest energy they can be. So it's spontaneous when you're at the top of the hill, like right here, you want to go down the hill. And you could go down either side. But in this case, the product is lower energy than the substrate. So you're going to want to go down the right side to be at a lower energy. So that's why you don't stay in the transition state. That's why you continue on to products, because the transition state is, or sorry, the product is lower in energy than the transition state. And molecules always want to go to low energy conformations. And so the other question is, what is the entropy penalty for staying in the transition state? So delta S. Really, the entropy penalty is paid upon first binding the substrate, right? Because if you're an enzyme, right, here's my enzyme. And I'm going to make it super simple. Let's say our substrate was water. And water has a ton of entropy, right? So the bonds of water will stretch back and forth. So this oxygen hydrogen bond, they stretch like a spring. So that's one freedom of movement they have. The hydrogens also move left to right together. So they are swinging back and forth. So they're swinging towards the oxygen. They're swinging towards each other. This oxygen also has 3D movement. And it's also spinning, right? So you can see all the different ways this oxygen can be arranged in space. There's a ton of different ways, right? Because it's spinning, it's moving, the bonds are flexing. All that is entropy, right? All that's chaos, so that's good. However, when that water molecule binds to the enzyme, it's frozen in place, right? It's now stuck. And that you're going from chaos, which is up here, to order, which is down here, which makes delta S negative. You have just decreased chaos by binding a molecule because it's no longer free to diffuse, no longer free to rotate, no longer free to stretch its bonds. However, we know binding is spontaneous. So if entropy is negative and binding is spontaneous, which means G is negative, Gibbs is negative, that must mean enthalpy is also negative. So the reason why we can pay this entropy penalty of binding water in a fixed conformation is that when we bind, we make new interactions between water and the enzyme, right? Where whatever water binds, there's probably some positive amino acids or hydrogen bonds just waiting for the oxygen, and either negative amino acids or hydrogen bonds waiting for the hydrogens. So you're making these nice new connections. When you make connections, you release heat into the universe. That's favorable. The universe wants that heat. And so we pay for the entropy by making new bonds and having negative enthalpy. So that's how that system works. That's how binding anything works. And the last question on the message board is general study tips for the exam. All right, the way I would study for this is I would go through the PowerPoints and look at all those questions we did in class and see if you can answer all those, right? In fact, what I might do if you have the time is just make flashcards of all of those questions. I mean, it might be a little too late to do that now with only 2 days left, but it's still possible. But you want to study those. After that, I would go back to the tests, and if I'll make sure that those are, I think those are open where you can see them. But I'll go through the test and see if you can re-answer those questions without looking at the answers and see if you can get them right or looking at the correct answer. So questions in class and then previous test questions. Then I would go to the study guides, make sure you can answer those questions, right? Just go through them. Can you answer them? If you can answer all the questions in class that we went over, all the questions we have on the test, all the study guide questions, you should be perfectly fine. I don't think there will be anything that I'll ask you that you haven't seen already. So that's what I would do. And then, of course, just general going over the slides, looking at the information you're not quite sure on, just brushing up on your weak points. And that is all the questions we had on the message board. So after going through that, does anyone want to ask any other questions for the final? Could I talk a bit more about plasmids? Sure. Let me look. Slide with the plasmids in it. Let me share the plasmid slide. All right, so a plasmid is circular DNA that's found in bacteria, and in normal bacteria, they usually have some kind of gene that's on there, some kind of resistance, and bacteria can pass them from one to another. For us, we use them to incorporate DNA into bacteria so we can make bacteria produce proteins for us. And so there's a couple of areas of interest of plasmids. One is this region with restrictive enzyme sites. And this allows us to use restriction endonucleases to cut open our plasma, insert our DNA, and then seal it with a ligase, right? So that's one region of importance of a plasmid. Another region is the Lac Operon system. Usually you want to be able to control when the bacteria make a protein. So the general concept is you grow bacteria with your plasmid in growth medium, LB, and you watch how cloudy the LB becomes, because that cloudiness directly correlates to how much E. coli you have in your LB medium. And you want to start protein growth when the E. coli are at their highest concentration before they start dying. Because you can plot this out where you have time and E. coli growth. And it kind of looks like something like that, where at the beginning where you have E. coli and LB and you're shaking it, they all just replicate. And then the population will just plateau where the solution can't handle any more E. coli. You're running out of resources. And then the E. coli will just die off because there's not enough food to go around. It's becoming too crowded. And so the E. coli will just start dying. So you want to make your protein express at the highest population before they start dying. And so you will usually add some molecule, in this case would be lactose, to start your protein growing. So that's what the Lac Operon system's for. And this ampicillin resistance is our selectable marker. Right. So the idea is we have our DNA in this plasmid, and we want to get this plasmid into bacteria. And getting bacteria, plasmids into bacteria is very low percentage. You know, you're looking at like for every 100,000 bacteria you have, maybe one of those will uptake your plasmid. You know, that's how low percentages we're talking about. And so when we try to put our plasmid in the bacteria, we want to make sure that we're only growing bacteria that have our plasmid in this LB media. So this plasmid also gives resistance to ampicillin. So we're gonna try to force our plasmid into the bacteria. Then we're gonna take that bacteria that's been exposed to our plasmid and put it on a plate that has LB agar, so again, growth media, but that agar also has ampicillin, also has an antibiotic on it. So any bacteria that did not get our plasmid is not resistant resistant to ampicillin, and they will die. Any bacteria that uptook our plasmid will be resistant to ampicillin, and they will grow into colonies, right? They will replicate on that media, and you'll see a little growth after like 12 hours. So basically, well, yeah, 12 hours. So basically what you do inside the lab is that you give the plasmid to the bacteria, you shake it around your test tube, you put your test tube, you take the bacteria from the test tube, put it on a plate, spread that around, put the plate inside of like a little bit of heating chamber that's like 37 degrees Celsius, leave for the day, come back in the morning and see if there's any spots on your plate. Because if there are growth spots, little bacteria colonies, that means, okay, that colony has my plasmid. You then take that bacteria, put it into a beaker full of LB, plus, in this case, would be ampicillin as well, just as another way to make sure nothing grows in that LB broth. By the way, if you don't know what LB is, and you've kind of been wondering why I keep saying LB, it's just food for bacteria, right? It makes bacteria grow, and it has everything it needs for bacteria to grow. So you just have a test tube full of this LB plus ampicillin, which is an antibiotic. So again, this is a way to make sure only bacteria with the plasmid grow. And yeah, that's a plasmid. Usually you want them under 10 kilobase pairs. Otherwise, they're too big to go into a bacteria easily. And they have to be big enough to have a selectable marker, some kind of protein control system, and all these restriction endonuclease sites so you can insert your DNA. Well, that's probably all you need to know about plasmids. Anything else? And if there are no other questions, looks like there are. Can you go over finding a peptide charges? So I already went over that earlier in this, so I'm not going to redo that question. Just go back when I post the video for this. Just go and look at that explanation, as yeah, I did go over about how to find charges at pH 1, 7, and 14 for peptides. Is the last homework assignment up? It should be. I can double check. I thought I put that up on Wednesday. Oh, okay. New system. Apparently, even though I put it up, even though I said have it, you know, so that you can see it, apparently I have to go and physically check a green check mark next to the assignment as well. Otherwise, you can't see it for some stupid reason. So now that check mark has been clicked, and you should see it. And this is all about, so the assignment is all about stuff that we talked about last week, all about like enzymes. So it's good practice for you for the final exam. Anything else? All right, since I'm not seeing anything else pop up, if you do have other questions while studying, feel free to shoot me an email. I'll be posting this, and I'll just post it right at the top of Blackboard. But otherwise, yeah, study hard. Good luck. And just final reminder, it opens, the final exam will open up on Wednesday, 12 a.m. and you will have anytime all day to take it at your leisure, 2 hours cumulative, and some new information. Yeah, otherwise, I guess I don't have anything else for you. So like I said, email me with any questions, and yeah, have a good rest of your summer. And those of you graduating, congratulations. Those of you going on to next semester, it's sad to say, next week, Thursday, first day of classes. So see people in person then. See you later.